This video is on my handwritten notes on molecular mechanism of aqueous humor outflow resistance. Now there is resistance to conventional pathway and there is resistance to unconventional pathway. The conventional pathway is through the trabecular meshwork, then juxtacanalicular tissue, Schlemm's canal, collector channels, then deep intrascleral plexuses and episcleral venous plexuses, draining into anterociliary veins and then superior ophthalmic veins and then into the cavernous sinus. That is conventional pathway. And unconventional pathway includes two types, uveoscleral and uveovortex. Uveoscleral is from the anterior face of the ciliary body into the suprachoroidal space and the choroidal veins. Then the second one is uveovortex. Uveovortex is through the Arves vessels, then ciliary body, and then into the suprachoroidal space and the vessels. Now, resistance to conventional pathway. Precise mechanism of resistance to conventional pathway is not known yet. But there is one study, and in that study what Grant demonstrated is 360-degree incision of trabecular meshwork, also known as trabeculotomy, that eliminated approximately 75% of the normal outflow resistance, but above 7 mm of mercury pressure, it is only about 50% reduction, and rest of the resistance comes from what? It comes from intrascleral outflow apparatus of channels. Another study in monkeys was carried out and it shows contribution of following structures to the aqueous outflow resistance. Trabecular meshwork offers 60 to 65% of the resistance. Inner 1/3 to half of the sclera offers 25% and outer 1/3 to half of the outer scleral channels, they offer 50% of the outflow resistance. When the intraocular pressure is high, that time why the aqueous outflow resistance is high? So it is because of the distension of the trabecular meshwork. How it happens? Because there is increase in the number of size of endothelial vacuoles and giant vacuoles. Ballooning of those vacuoles, it causes the blockage of Schlemm's canal and collapse of Schlemm's canal, and that causes increase in the intraocular pressure. Now, whenever there is distension of the Schlemm's canal, there is decrease in the aqueous outflow resistance. Why it happens? Because the trabecular meshwork is a three-dimensional structure and made up of the diagonally crossing collagen fibers, and it has an effect on this trabecular meshwork cells. And distension of the Schlemm's canal causes opening of this trabecular meshwork spaces, and that's why it causes decrease in the aqueous outflow resistance. And also, whenever there is posterior displacement of the lens or tension on the choroid, that causes tension on the trabecular meshwork also, and that causes opening of the Schlemm's canal also, and it increases the aqueous outflow facility, thus having an IOP lowering effect overall. Now, the pattern of aqueous humor circulation within the Schlemm canal is not fully understood, but there are certain things from which we can conclude certain things. For example, in inebriated human adult eye, it showed aqueous humor cannot flow more than 10 degrees within the canal. In infant eyes, there was less resistance to circumferential flow. And studies of segmental blood reflux into Schlemm's canal, from that we can imply that canal is normally entirely open and there is a circumferential flow. Other perfusion studies using a tracery elements have shown there is relatively free flow through the trabecular spaces and the juxtacanalicular tissue, and it means there is less resistance offered by these structures, which are trabecular spaces and juxtacanalicular tissue. But when they reach the inner surface of the inner wall of the Schlemm canal, the resistance is encountered and the free flow is stopped there. so indicating that significant amount of resistance is offered by the inner lining of Schleim's canal. Okay, so main resistance imparted by the endothelial lining of the Schleim's canal. Histological changes in the aging eye in aqueous outflow pathway. So there is reduction in the size of the Schleim canal, narrowing of intercellular spaces, increase in extracellular material, especially electron-dense plaques near juxtacanalicular tissue that is associated with the ciliary muscle tendon inserting on the scleral spur with age. In chronic open-angle glaucoma, there is marked loss of trabecular meshwork cells, fusion, thickening of trabecular lamellae, that is trabecular beams, and increased electron-dense plaques in extracellular matrix that adhere to the sheets of elastic fibers and their connections to inner wall of endothelium. In steroid-induced glaucoma, what happens? There is increased fibrillary material that stands for collagen type 4 in the subendothelial region of the Schleim canal. In pigmentary glaucoma, what happens? Trabecular cell loss is more prominent than in chronic open-angle glaucoma. And there is overload of pigment granules to phagocytose and clear them of the trabecular meshwork. So, and also there is collapse of Schleim's canal, disorganized cribriform regions seen in the pigmentary glaucoma. Now, extracellular matrix. Resistance at the level of the extracellular matrix of the trabecular meshwork and the basement membrane. Extracellular matrix, it is important to regulate intraocular pressure. Its composition is important to regulate the intraocular pressure. It is made up of collagen, that is fibrillar and non-fibrillar collagen, elastin containing microfibrils, matricellular components, structural organizing proteins, glycosaminoglycans and proteoglycans. Now the state of components in the extracellular matrix, it is dynamic and there is constant turnover and remodeling taking place in the extracellular matrix, and that is in response to the intraocular pressure stretching, and it occurs through cell adhesion proteins, cell surface receptors, associated binding proteins, certain cytokines, growth factors and drugs. Now related studies to this are first in human eyes and then porcine eyes. What they have observed in human porcine eye studies, they carried out this study with beta-xyloside and chlorate. Beta-xyloside is competitive nucleation point for addition of disaccharide units, and chlorate is inhibitor of sulfation. So both this beta-xyloside and chlorate, what they do, they cause disruption of biosynthesis of glycosaminoglycans, a component of extracellular matrix, and this way they increase the outflow facility, that they decrease the outflow resistance and increase outflow facility, decreasing the intraocular pressure. Now, tissue plasminogen activator is a normal component of the aqueous humor, and what it does is it alters the glycoprotein content of the extracellular matrix and it causes the fibrinolysis, and that is favored in trabecular meshwork and endothelium of Schleim's canal. Overall, normally it is favored fibrinolysis as a protective mechanism against the obstruction from fibrin and platelet formation. So when there is fibrin platelet formation, it can obstruct the trabecular meshwork and endothelium of the Schleim canal. So this tissue plasminogen activator that is normally present in the aqueous humor, it has got the protective role against these clots. Also, it alters the glycoprotein content of the extracellular matrix to reduce the outflow resistance. So tissue plasminogen activator has a very protective role in reducing the aqueous outflow resistance. Now, glucocorticoid mechanism. Glucocorticoid, it causes inhibition of endogenous prostaglandin synthesis. Then it has got direct effect also on the extracellular matrix, metabolism and cytoskeleton. And glucocorticoid receptors are demonstrated in human glucomatous eyes, non-glucomatous autopsy eyes and cultured human tubercular cells. Now myocilin, TIGR expression. Myocilin expression is increased in tubercular meshwork in response to dexamethasone according to some studies. But in patients with steroid-induced glaucoma, they don't have myocilin mutations. So, and also we have seen previously, myocilin is found in the normal aqueous humor, but it is absent in certain forms of glaucoma. So, in patients with steroid-induced glaucoma, they don't have myocilin mutations, and myocilin expression is increased in tubercular meshwork in response to dexamethasone according to some studies. And it has a role in juvenile glaucoma. The details are not given here, you will see later on in childhood glaucoma. Now, cellular and cytoskeletal mechanism. Tubercular cells have actin, vimentin, and desmin. These are cytoskeletal proteins or cytoskeletal filaments. These have contractile phagocytic pinocytic properties. These impart contractile phagocytic pinocytic properties to the cell. Now, with aging, there is tubercular cell damage and degeneration. Also, it is seen in pigmentary dispersion glaucoma, pigmentary glaucoma. Now, this progressive accumulation of the damaged protein and misfolded proteins in tubercular meshwork when there is damage to the tubercular cells. Also, in pigmentary glaucoma, what happens? Lots of pigments are there, so there is overload on the cells to digest and to remove these pigments and the debris. So what happens? These damaged proteins and pigments and misfolded proteins, they accumulate in the tubercular meshwork. Then there is increase in the outflow resistance and increase in the intraocular pressure. In monkey eyes, they have observed with cytoskeleton, EDTA and H7, which disturb the microfilaments. There is alteration in the histology of the tubercular meshwork and Schlemm canal, and that causes reduction in the aqueous outflow resistance. So they lower the intraocular pressure, cytoskeleton, EDTA and H7 in monkey eyes. Now there are sulfhydryl reagents. Now these sulfhydryl reagents, these are iodoacetamide and ethyl malimide ethacronic acid, and they have the receptors or they work at the level of endothelial membrane of Schlemm canal. They alter the cell membrane sulfhydryl group here, and they increase the aqueous outflow. That is, they decrease the aqueous outflow resistance and decrease in the intraocular pressure. Now, another mechanism of sulfhydryl groups, for example, glutathione. Glutathione, we know that it is detoxifying molecule. It reduces the oxidative stress. And this glutathione, it modulates the aqueous humor outflow. It is involved in hydrogen peroxide, which is the normal constituent of aqueous humor. Hydrogen peroxide increases the oxidative stress, and the glutathione is involved in reducing the oxidative stress by this hydrogen peroxide. Now, studies which support the glutathione protective role is demonstrated with the study in calf tubercular meshwork. Calf tubercular meshwork, they have studied the sulfhydryl component glutathione and enzyme glutathione peroxidase. They catalyze the reaction between the glutathione and hydrogen peroxidase. That is the detoxifying reaction, detoxifying the H2O to hydrogen peroxide. And this way they protect the tubercular meshwork cells, and they protect the tubercular cells from the harmful effects of H2O. Because once the tubercular meshwork cells are damaged, they cause the fusion and hyalinization of the tubercular lamellae or tubercular beams, thickening of the beams. Also, the misfolded and damaged proteins, they accumulate in the tubercular meshwork, and there is increase in the aqueous outflow resistance. So this was all about the aqueous outflow resistance. Now here I want to combine these notes from Schill's glaucoma with notes from Wolf's anatomy, in which histological changes are observed in tubercular meshwork and aqueous outflow pathway with the aging and open angle glaucoma. So, in the aging eye, there is increased thickness of the tubercular sheets, that is 2 to 3-fold, and it is because of the accumulation of the curly collagen in the cortical zone and core of the beams and basement membranes. Then there is decreased collagen associated proteoglycan, which is chondroitin sulfate and also tomatin sulfate. Peripheral microfibrillary component of elastin becomes obscure. There is decrease in the number of tubercular cells because of the degenerative process in aging. There is depletion of cellular covering of tubercular beams, and there is subsequent fusion of tubercular beams. This process is also known as hyalinization of the meshwork. It leads to consequent reduction in open spaces of meshwork, development of resistance to aqueous outflow, and increase in the IOP. In the pericanalicular region, the cell loss, endothelial cell loss occurs. Then there is accumulation of 55 nanometer banded component that ensheaths the collagen, fibronectin, and elastic fibers. There is decrease in the content of hyaluronic acid in the pericanalicular region, and increase in the level of fibronectin type 6 collagen, thrombospondin, and there is decrease in the level of laminin in the pericanalicular region extracellular matrix. But there is accumulation of laminin beneath the endothelial lining of Schlimm's canal. In primary open-angle glaucoma, similar but exaggerated changes are seen, along with the paucity of giant vacuoles in the endothelial linings of Schlimm's canal. Although some investigators have disputed this, there is attenuation of Schlimm's canal that is seen as an early event. It may be an early event. There is also decrease in the diameter of collector channels, but it is not apparently early in the disease. So that was all about the molecular mechanism of aqueous outflow resistance. Hope you have understood this. Thank you so much.